We're very excited about this cancer discovery paper. We have found that CDK6, which is a cell cycle kinase, has a second life as transcriptional regulator. You may know that there are drugs against CDK6 kinase. Palbociclib made it through on the market. It's a very important drug. It's a CDK4/6 inhibitor. And there are a lot of clinical trials going on, in particular also in AML. And we found that CDK6 is also interfering not only with cell cycle, but also with p53. And we describe in our manuscript that CDK6 is counteracting p53 responses. So when you block CDK6, p53 is not regulated. And this has important consequences, and it could be a bless or it could be a curse. That has to be tested in the future. On the one hand, if we block CDK6, we could promote p53 responses and drug responses could be stronger and cells could be killed more effectively. But on the other hand, it might also be that it promotes p53 mutations in pre-leukemic clones. So it has both sides, and future needs to tell us what is going on and how we make best use of these new drugs, the CDK4/6 inhibitors. We are working with mouse models, so we are having mice that lack either CDK6 completely or that have a point mutation in CDK6, and CDK6 is there, but it's not kinase active, and then we have wild-type mice, and we do different leukemia models, and we sort of mimic the inhibition of CDK6 in our mice, and we also treat wild-type mice with palbociclib, and this is how we came to our conclusion and reached our conclusions. And of course, nothing works nowadays without ChIP-seq analysis, RNA-seq analysis, phosphochromosome analysis. So we use the whole entire modern omics technology to study what is going on. We found that CDK6 is actually phosphorylating two transcriptional regulators, SP1 and NFI. And that's the mechanism how it interacts with p53. And this phosphorylation by CDK6 is a prerequisite for p53 to dock on and to induce this counter-regulatory set of genes that is blocking p53 functions. Whereas the pro-apoptotic genes like PUMA and NOXA are direct targets of p53 and do not require CDK6. So the whole balance, if we block CDK6, is shifting towards the pro-apoptotic side of p53, and that's the underlying mechanism. The interesting thing is it's approved, FDA approved for breast cancer treatment and other treatments. Their clinical trials are ongoing in AML and ALL. And the interesting thing is that a lot of the hematopoietic malignancies have very high CDK6 levels. CDK4, which is considered an identical twin, stays low, and CDK6 is upregulated. So that prompted us, like 10 years ago, to look closer at these identical twins, and it turned out that CDK6 has this additional function as transcriptional regulator that is not shared in hematopoietic cells by CDK4, and that makes CDK6 such an interesting and unique model system. And we also found that some of the transcriptional functions of CDK6 are kinase dependent and some are kinase independent. So some of them will be blocked by palbociclib and others will not. We have verified that the transcriptional response that is counteracting p53 is also present in human malignant diseases. We have verified it in ALL, in AML, and in CML. And we could also show, together with a group in Harvard, Ben Ebert and Donna Newberg, that patients that have 7q deletions, which means that they have only one allele of CDK6, have a higher probability to have p53 mutations. So the data that we obtained in mice seem to be reflected in human patients, and I think the paper is now making people aware that this is a certain risk but also a blessing that could be exploited and should be further explored.